Hello students, this is Dr. Fazan Mirza. What I'm doing here in this video, I'm sharing with you some questions, some recent questions from the chapter of infectious diseases. So I've taken these questions from 2023. So let's get on with this. The first question that I'm displaying to you here in this particular video is the one which is here from May-June 2023, variant 22, and this question asks you regarding something like trypanosoma, an organism, unicellular organism that causes infectious diseases known as sleeping sickness. Insects known as sexy fly pass this organism from infected people to uninfected people when male and female sexy fly feed on human blood. The transmission electromicrograph of human blood taken from the blood, this organism is shown to you. So you can see this diagram here. In this diagram, you can see this organism is having a nucleus. And this is having a nuclear envelope, and this must be the nucleus. And you can see the rough yards and the mitochondria as well inside. And there's some extension here. So let's move on. Draw an arrow indicate the location where ribosomal RNA is made. We know ribosomal subunits are produced in the nucleoli, and this structure is the nucleus. Inside this is the nucleoli, so we'll just mention where this is occurring. With reference to the figure, explain how the structure labeled X provides evidence this organism is motile. So let's look at this structure X. This structure X here, you can see this diagram. This diagram shows to you that this organism is having this outer part, and this ends here. So this is where the cell ends. But at the same time, this is something also present attached to the structure of the cell. So this must be something which is like a flagella, and this must be providing motility, but it is cut in cross section, not along the length, so you don't see it very long, rather you see it just cut as a separate section. And inside you can see these microtubule triplets which are present there. This typical 9+2 arrangement that we study, we can see this here. So the answer that we mentioned here is the 9+2 arrangement is visible on the structure X. There's appearance of the structure of the cylindrical and the line outside the cell, so it's providing motility to the cell. So that's what we can expect. We can actually mention here. With reference to figure 4.1, explain whether this organism is a eukaryote or a prokaryote. Definitely it's a eukaryote, and why? Because the true nucleus can be seen together with the nucleoli, mitochondria, the endoplasmic can be seen. This is pretty large organism for being a prokaryote. So it's just about 1 to 5 micrometer per prokaryote. A typical prokaryote is 1 to 5 micrometer, and you can see this is pretty large. This part is 1 micrometer, so you just keep on adding. So this is very large organism. Plus, there's a 9+2 arrangement of microtubule, which is covered by the cell membrane. So the 9+2 arrangement being covered by the cell membrane is a feature of a eukaryotic cell. So yeah, we can mention this as well. State the name for the malarial pathogen, malarial plasmodium, how the organism causes disease. We know it's a pathogen. Name one of the species. So out of the four, you mention any one. I've mentioned falciparum here. You can mention malaria or ovale or any one of the four that you have studied. Outline the similarity and difference between the modes of transmission of malaria and sleeping sickness. So sleeping sickness and malaria, they both are vector-borne diseases, vector-caused diseases. In fact, not vector-caused, vector-transmitted disease. So vector is there for both malaria and female anopheles mosquito sleeping sickness by the flies. So that's the similarity. Both feed on human blood. That's a similarity as well. But we have to mention the differences along with this. Both are not caused by the same mosquito or by the same fly. So that can be the difference that we can add here. So this was one question that I had taken. Let's move on to another question from the same series of October, November, and let me just look for this question and we can move on. So this question here is HIV/AIDS, October, November 23, variant 21. Some people who are infected with HIV develop HIV/AIDS. Figure 4.1 shows the number of people affected by newly infected HIV new infections in 2018 across the world and the percentage changes in the number of new infections since 2010. So this is the chart shown to you. In various regions, you can see in A, new cases are these many, 12% decrease. In B, there's a 29% increase. This shows increase. In C, 9% decrease. In D, 10% increase. So this shows increase, this shows increase. E, 28% decrease. F shows 13% decrease. G shows 7% decrease. So I have just annotated what I can see here. These two areas, they show increase. Largest decrease is shown by E, just using the numerical values here, because this is what the relevance of the numerical values. We actually can compare one thing against the other. The smallest decrease can be seen in G. And I just mentioned all the regions where there we can see the decrease here. State the full name of the HIV pathogen, something that students get wrong. Human immunodeficiency virus. So nothing else but these three terms. Using the only data provided here, you need to just state what can be concluded. So whatever I mentioned here, these all these things can be included in the conclusion regarding this data. People who develop AIDS have fewer T lymphocytes, and that for destroying pathogens, this makes them more susceptible to TB. Explain why people with HIV are more likely to develop TB. So TB is an opportunistic infection. We have learned this. It affects the people who have a weakened immune response. Helper T cell count in HIV/AIDS patient is reduced already because that's where the pathogen lives. This reduces the cytokines too. So B cell to plasma cell proliferation occurs which in less, we can say at a lower rate. Fewer antibodies are produced, memory cell count is low too. So even if they are BCG vaccinated, still they get infected with the pathogen. So you need to get 4 points there. Then they take this question to the transported mammals. That's what they do. They integrate the questions together with other chapters. So a person with HIV infection develop heart condition called cardiac tamponade. The pericardium is a thin sac surrounding heart. This is the new information provided to you. You can see this is the heart structure, and this is the pericardium sac. This is some this is a layer which you have not studied unless you know what pericardium is from something that you have studied. Otherwise, we don't study pericardium in AS. So they have shown you a healthy heart, and then when a person develops cardiac tamponade, the pericardium starts to fill with blood. This prevents ventricle functioning efficiently. So you can see this pericardium is filled with blood. So, and these arrows are there to tell you there is a pressure in the pericardium. So, how would it affect heart functioning of a person with cardiac tamponade and a healthy heart? People with cardiac tamponade experience variety of symptoms, including low BP and increased breathing rate. Suggest they're asking you to suggest suggest why a person with cardiac tamponade would experience symptoms of low BP and increased breathing rate. So, pericardium in tamponade exerts pressure around the ventricle. This reduces ventricular filling. Ventricles are not fully filled, so less blood goes into each ventricular systole. Less blood goes into the lungs for oxygenation. So, blood circulated when blood is not fully oxygenated, still carry a lot of carbon dioxide. So, patient starts to breathe faster. That's what you can just mention in this answer. Let's move on and pick another question from the same series. October 2023, variant 23. This is malarial pathogen P. falciparum, and this is shown to you inside the red blood cell. You can see this is the outline of the red blood cell membrane. You can see the membrane appears a bit fragile. It's not exactly the disc-shaped cell anymore. Inside the cell, you can see there's a parent cell and this is a cell dividing and splitting and budding of new cells. And you can see nucleus inside every pathogen here. So we know that how the falciparum is basically a protoctist. It's a eukaryotic cell infecting the red blood cell. With reference to figure for 1.1 suggests how the presence of P falciparum affect the red blood cells. So as the pathogen divides the RBC, it uses up the hemoglobin for new as a your source of nutrient for its growth. The RBC swells and loses biconcave shape. The cell membrane eventually bursts. Next, they're asking you when P falciparum divides, they are asking to you what there is unequal division of cytoplasm to form small genetically identical daughter cells. Outline the events that occur in the cell P falciparum to form daughter cells shown. Unequal cytoplasm division is occurring. They have mentioned the small genetically identical daughter cells are produced. So as plasmodium grows inside RBC, it spends the interface by producing more RNA and proteins. We include cell cycle there. In S phase, it will replicate its DNA. In M phase of mitosis, prophase, metaphase, anaphase, telophase follow occur, and cytokinesis occur. Daughter cells are produced, and new cells bud off, and the parent cell still is there, and the number of chromosomes are the same, and the cytoplasm is there as well. So you can see this budding off here. So if it's visible in the cell, and the parent cell is still there, and it's producing unequal number of copies, so we need to include the term of budding here. This is where they took the question to assessment objective 2. Next, they have given to us information about some vaccination and how this control group basically vaccine occurred. And they are simply asking us to vaccine stimulate immune response, explain how antibodies will reduce the spread of malarial pathogen through bloodstream. We know that if antibodies are there, the variable region will be complementary to the antigen. They will bind to the malarial pathogen. Malarial pathogen cannot penetrate to the red blood cell and gets marked for destruction by neutrophils, something that you learn in the chapter of immunity. Then they ask you about vaccination. So some vaccination programs have been more successful than others. Discuss the factors that contribute to success of vaccination program. So what makes the vaccination program successful? You have to add 5 marking points here. I feel this question is a bit difficult for the students because you add a lot of general information here. So vaccination program must be included to ensure that they include the people at risk of getting infections or 95% coverage, so herd immunity can be reached, and this will help protecting the people who are not vaccinated. Children should be vaccinated early in life. Awareness program should be arranged for the parents regarding benefits of vaccination. Vaccine should not be very expensive and be stable enough for long-term storage and usage, and transportation. Authorities should make plentiful supply of vaccines available for the people at risk, so that will eventually get you good marks. I would recommend that you attempt these questions yourself now and try to see how much this video was helpful for you in assisting you with this topic. That's it from my side. Thank you so much.